Red light therapy, a potential ally in reducing blood clot formation. Introduction. Blood clots, medically known as thrombi, are significant contributors to life-threatening conditions such as heart attacks, strokes, and pulmonary embolisms. Traditional preventive measures encompass anticoagulant medications and lifestyle modifications. Emerging research from the University of Pittsburgh School of Medicine and UPMC introduces an innovative approach, red light therapy. This non-invasive method may significantly reduce the risk of blood clot formation, offering a novel avenue for cardiovascular health maintenance. The science behind red light therapy and thrombosis. A study published in the Journal of Thrombosis and Hemostasis explored the effects of long-wavelength red light on clot formation in both murine models and human data analyses. Mice exposed to 12-hour cycles of red light over 3 days exhibited a remarkable 5-fold reduction in clot formation compared to those subjected to blue or white light. Importantly, variables such as activity levels, sleep patterns, and body temperature remained consistent across all groups, underscoring the specific impact of red light exposure on thrombogenesis. Further analysis of over 10,000 cataract surgery patients revealed that cancer patients who receive blue light filtering lenses, which reduce blue light exposure by approximately 50%, had a notably lower risk of developing blood clots compared to those with conventional lenses. This finding is particularly significant given the heightened thrombotic risk associated with cancer patients. Mechanisms underpinning red light's antithrombotic effects. The protective effects of red light therapy are attributed to several physiological mechanisms. Reduction in inflammatory response. Red light exposure is associated with decreased levels of pro-inflammatory cytokines such as interleukin-6 and monocyte chemoattractant protein 1. This attenuation of inflammation contributes to a lower propensity for clot formation. Modulation of neutrophil extracellular traps, nets. Nets are web-like structures released by neutrophils that trap pathogens but can also ensnare platelets, facilitating clot development. Red light exposure has been shown to reduce net formation, thereby mitigating this risk. Enhancement of fatty acid production. Increased fatty acid synthesis under red light exposure leads to reduced platelet activation, a crucial step in the clotting cascade. Notably, these effects are mediated through the optic pathway. Experiments with blind mice demonstrated no change in clotting patterns upon red light exposure, indicating that visual perception of light is essential for these therapeutic benefits. Potential applications and future directions. The implications of these findings are profound, suggesting that red light therapy could serve as a cost-effective, non-invasive strategy to reduce thrombotic events. Researchers are developing specialized devices, such as red light emitting goggles, to facilitate controlled exposure for individuals at elevated risk of clot formation. While these preliminary results are promising, further clinical trials are necessary to validate the efficacy and safety of red light therapy in diverse populations. Understanding the precise biological mechanisms will also aid in optimizing treatment protocols and potentially integrating this therapy into standard preventive cardiology practices. Conclusion. Red light therapy emerges as a compelling adjunctive approach in the prevention of blood clots, leveraging the body's response to specific light wavelengths to modulate inflammatory and coagulative pathways. As research advances, this modality holds the potential to enhance cardiovascular health and reduce the global burden of thrombotic diseases.